These are not leaves. They are highly engineered biological kill rooms deployed by carnivorous plants to extract living flesh. The waterwheel plant. In the shallow, nutrient-dead waters of Eurasia, this rootless anomaly faces a severe physical problem. Chemical attractants diffuse instantly in water, leaving the organism with an impossible biological hurdle, securing erratic aquatic prey before they escape. The solution is a masterclass in localized hydrovacuum engineering. Highly sensitive trigger hairs mandate stimulation in rapid succession, a physical failsafe to prevent energetic bankruptcy from misfires. Once the mechanical threshold is breached, a hydraulic snap closes the lobes in 10 to 20 milliseconds. This ranks among the fastest biomechanical movements in the plant kingdom. The rapid closure forces immense water displacement. It operates exactly like a pressurized pneumatic hatch detonating inwards, utilizing surrounding fluid dynamics to violently suck mosquito larvae into an airlock before the jaws hermetically seal. The raw efficiency of the capture is staggering to witness. The COBRA LILY. How does a terrestrial structure force flying insects to surrender their momentum and walk into a liquid grave without ever actively snapping shut? Rising from the serpentine bogs of Northern California, this structure is not a leaf, it is a terminal. To secure aerial prey, the mechanism relies entirely on spatial disorientation. The biological hood features fenestrations, translucent patches lacking chlorophyll that act as false windows. The target exhausts its battery, flying upward into solid plant tissue, thoroughly disoriented by shifting light. Gravity inevitably claims the exhausted prey, pulling it down a shaft lined with downward-pointing microscopic hairs. This architecture acts as a mechanical ratchet, physically preventing upward movement. It functions identically to a casino designed with opaque exits and disorienting ceiling lights, ensuring targets burn through all internal resources before inevitably collapsing into the vault. At the base of the shaft, the plant deploys no native digestive enzymes. It relies entirely on a highly concentrated microbiome of symbiotic bacteria to chemically liquidate chitin and soft tissue, silently processing armored beetles into harvestable nitrogen. The CAPE Sundew. In the scrublands of South Africa, a fly descends toward a glittering drop of morning dew. It is a fatal miscalculation. The droplet is an industrial-grade viscoelastic polymer. The moment the insect lands and attempts to pull away, the mechanical resistance of the glue exponentially increases. Struggle is the precise catalyst for its own destruction. As the insect burns through its energy fighting the adhesive, the physical disruption triggers a response. Asymmetric cellular growth forces the entire leaf to slowly coil around the living prey. The mechanism operates identically to a hydraulic vise. There is no neurotoxin to paralyze the target. The thick fluid physically clogs the breathing spiracles of the insect. It suffocates while entirely conscious. As the insect asphyxiates, the plant deploys esterases and proteases to chemically liquefy its soft tissues. This localized death spiral is brutal but stationary. Deep in the rainforest, a botanical anomaly weaponizes this exact adhesive terror, only temporarily, using it as a biological rocket booster before jettisoning the hardware entirely. The VENUS FLYTRAP. Stationary glue traps fail in environments with constant environmental disruption like heavy rain. In the coastal bogs of the Carolinas, how does an organism prevent catastrophic energy bankruptcy from a false alarm, ensuring it only snaps shut on a living target? The answer is an absolute biological timer. A spider crawls into the open rosette. Inside, microscopic trigger hairs act as a memory system requiring strict two-factor authentication. The spider brushes a single hair. An invisible countdown starts. A second mechanical stimulus must occur within a 20-second window. If the spider freezes, the timer resets. The organism simply waits. The moment the spider breaches the second hair, the trap deploys stored elastic energy. The leaf geometry violently flips from convex to concave in under 100 milliseconds. The spring-loaded biometric vault initiates total lockdown. Post-capture, the architecture transitions into an external stomach. It hermetically seals around the captive to lock out ambient oxygen. A 10-day enzymatic liquidation phase begins. The chemical balance is highly restricted. Overloading the vault with dense biological matter collapses the system. If it captures prey too large, or if fed raw mammalian meat, the enzymes fail. Lethal putrefaction rots the trap from the inside out. The fanged pitcher plant. We leave the bogs of the Carolinas and enter the peat swamps of Borneo. A lone foraging ant navigates the slick upper rim of a colossal botanical vault. To access highly concentrated nectar, it must perform a lethal tightrope walk. It hangs completely upside down from two downward-pointing structural fangs, balancing precariously over the exact center of an open abyss. Gravity constantly pulls at it. A microscopic slip of a single claw breaks its traction. The ant drops. It is a fraction of a second of absolute free fall into the biological reservoir below. The fluid waiting at the bottom is not water. It is a viscoelastic, non-Newtonian polymer swamp. As the ant hits the surface, panic sets in. It violently thrashes to swim. This sudden explosion of movement is a fatal error. The mechanical drag of the liquid exponentially increases with every struggle, instantly turning the fluid into biological concrete. The pure survival instinct of the insect seals its own execution. Escape is physically impossible. Left alone, the accumulating dead matter would overload the system, causing the plant to rot from the inside out, exactly like the Venus flytrap. To survive its own lethal efficiency, the organism operates a mutualistic cartel. A colony of specialized mercenary ants lives permanently inside the plant structure. These insects dive directly into the hazardous fluid to retrieve drowning prey. By stripping the excess biological load, the ants prevent the vault from collapsing under its own gluttony. It is a perfectly organized, self-cleaning execution chamber. The yellow pitcher plant. Structural traps and biological syndicates require time and heavy maintenance. In the pine savannas of the southeastern United States, how does a predator rapidly subdue aggressive, winged targets without relying on viscous fluids, moving parts, or mercenaries? The answer is targeted chemical warfare, a quiet poisoning. A wasp lands on the upper rim of the tube. It feeds on the abundant surface nectar. This bait is not merely a lure. It is laced with coneine, a potent alkaloid neurotoxin, identical to the active lethal compound in poison hemlock. Ingestion triggers immediate central nervous system depression. The wasp suffers rapid motor function paralysis. The rim of the trap features microscopic, unidirectional ridges that become virtually frictionless when lubricated by ambient humidity. Stripped of mechanical control, the paralyzed insect simply slides down the slick funnel. Inside the shaft, rigid, downward-angled hairs act as a mechanical non-return valve. Any desperate attempt at upward traction by the failing insect is physically blocked. It drops to the base. There is no pool of water to drown in. The tube is entirely dry. The kill relies strictly on neurotoxic paralysis, followed by localized chemical dissolution at the bottom of the shaft. The giant pitcher plant. Deep on the slopes of Mount Kinabalu rests a 3.5-liter biological cauldron. It perfectly matches the visual blueprint of a man-eating monster from cheap science fiction. While entirely harmless to a human observer, its physical scale is staggering. A mountain tree shrew cautiously approaches the rim. One microscopic slip into the fluid below means a slow, inescapable dissolution by potent proteases fully capable of stripping mammalian cartilage from bone. The shrew steps onto the edge of the abyss. But the trap does not trigger. The mammal is not prey. It is a biological contractor. The plant actively feeds the shrew, producing immense amounts of highly caloric exudate on its lid. The transaction is brutally pragmatic. The plant provides premium fuel, and in return, the occupied shrew defecates directly into the biological reservoir below. Extreme nitrogen deficiency in montane soils forces this organism to abandon erratic insect captures. It requires stable nutrient loads to survive. It operates as a structural toll booth, strictly exchanging calories for concentrated chemical waste. The occasional drowned vertebrate is merely an accident. The true masterpiece is the systematic, highly engineered harvesting of mammalian excrement. The tropical vine trap. Patience in passive biological vats are effective, but only if you have time. If an organism is locked in a brutal race for canopy sunlight, it requires an immediate, explosive influx of nutrients. How does a standard 50-meter woody liana suddenly weaponize itself to slaughter thousands of insects, only to completely erase the evidence of its crimes once it reaches the sun? A juvenile vine in the West African rainforest faces a severe survival problem. The biological race for canopy sunlight requires vertical growth. It needs a concentrated nitrogen booster that its standard roots cannot extract from the leached soil. The organism executes a temporary radical shift. It essentially becomes a transient serial killer. Prior to its rapid ascent, it alters its structure to produce specialized leaves dripping with what appears to be shimmering nectar. It is, in reality, an industrial-grade adhesive. An entire swarm of insects descends on the false bait. The moment they land, they are pinned. Panic spreads across the leaves. Thousands of insects thrash violently against the resin to escape. This sheer physical struggle is the exact mechanical trigger the vine requires. The force of their panic tears open microscopic glands across the leaves, flooding the trapped, exhausted insects with highly corrosive digestive acids. The harder the swarm fights, the faster the acid flows. Within hours, the frantic thrashing stops. The execution reduces thousands of bodies into a pure liquid nitrogen payload. The vine quietly drinks the graveyard. This extracted fuel hits the biological system like raw adrenaline. Fueled by the carnage, the vine triggers an explosive vertical ascent, violently pushing its way through the dark understory. Then comes the breakthrough. The vine breaches the high canopy. Unobstructed sunlight finally hits its upper structures. The biological starvation crisis is over. With its ultimate target achieved, the organism completely terminates its carnivorous mechanics. The lethal execution chambers wither and detach, dropping to the forest floor. The predatory hardware is jettisoned entirely. What remains in the canopy is a standard, sterile, woody vine, peacefully photosynthesizing in the sun. It is a completely harmless plant, quietly hiding the mass grave that funded its ascent.